अगर 40 का ब्रेक देती तो 45 का ब्रेक देती है, 9 का डेज हम लोग को क्वेश्चन मार्क से ना बात है ना पढ़ाई कर जाओ क्वेश्चन मार्क का सिस्टम अभी नहीं पढ़ना बोलती हूँ हम लोग पढ़ते नहीं उससे बस सॉल्व करते हमें सिर्फ सॉल्व करना हम लोग को देंगे करना सर बट हम लोग को उससे पढ़ना नहीं बस बच्चे होते से पेपर बुक नोटबुक पता नहीं क्या पढ़ते रहते हैं नहीं तो मार्क्स देना कितने लोग से बात करते हैं सीएसके चूज अच्छा नहीं पता सीएसके चूज बोल अभी सीएसके चूज बोल हो गया सपोर्ट हो गया 20 on 0 हो जाएगा भाई को आंसर कॉस्ट पे है केवल नहीं सारा ये तुम्हारा भाई पिछले साल आ गए एक बार वो उनको आगे ज्ञान नहीं हमारा अब पहले अभी ठीक नहीं करना दो वाले भी दिए मैंने दूसरा एक ही दिया बट जब से सीएसके और फिक्स तो ये जब से सीएसके वन ऑलरेडी हो गया
मैं भी तो अग्री डोमिनेटिंग करती हूँ हाँ
वॉल्यूम में ज़्यादा पड़ रहा है और वो लास्ट चेक ठीक है फिर अभी टर्म 2 में भी ठीक है आई एक्सपेक्टेड मोर मेरे को थोड़ा बड़ा मेरे को हां वही क्वेश्चन नहीं था
आदि फर्स्ट सेट है हमारे कितना पॉइंट है 2 पॉइंट है 15 तो दाल क्या है वही पॉइंट बता दे भाई एक्चुअली टाइम इसीलिए सेटअप में ना मुझे सब जो भी करना है जैसे सीमा चला के मेरे सेंटर ऊपर क्या बहुत वॉलीबॉल खेल रहे हैं भाई मैं आपके ये सेंटर है बोलते हैं मैं आपके खेल रहा था शॉपिंग के बाद लेफ्ट विंग राइट विंग क्या लेफ्ट विंग गर्ल्स फर्स्ट टाइम सीओ में बॉल सीओ राइट स्पेशल लिबेरो और तो उसने ठीक ही करा हां लास्ट सिलेक्शन स्पेशल तो सारे अच्छा करे एक रीडिंग ओवर मैं रीडिंग अभिप्राय से फ्रेंडशिप नहीं सारा मैं द्वारा से ठीक करना सॉरी लेकिन वो मैं पता सिंपल से नहीं और नहीं इतना अच्छा सिलेक्शन की पहले मुझे नहीं पता थी कि जीरो आ रहा मैं शॉपिंग और किधर हमें तो 15 से पहले पता था वॉलीबॉल का वो एक फर्स्ट सेकंड से तक सेकंड कोई भी अगली से लेफ्ट दिखा उस लेवल पे मेरा लेफ्ट की उर्वी से फर्स्ट से अच्छा हुआ पहले कोई से दोनों एंड टर्न टू ऑर्डर वेरी कौन है अभी ये तो समझा बेटा ये क्या बोल रहा है पूरा टाइम एक बार भी स्विच करो अबे लेकिन आपने हाउस में हाउस ये सारी का एक बार भी खेला गया एक समान सब अरे आगे हो क्या एक बार भी अंदर नहीं आया और अली ने अभी को काली वो एकदम पसंद आ जाता है सेलिना पड़ेगी ना देखो भी आए अरे भाई मैं ओए सर आरुष ना अपने ना शेयर पर सभी को बहुत अच्छा है हम लोग जीत रहे थे क्यों सब नहीं करे हम लोग चेक आउट पढ़ने से उसको हमने जो भी अबे आदित्य तो इससे क्यों कर रहा है इसे कुछ करा ही नहीं उसने कुछ नहीं कर रहा और 10 मिनट चाहिए और 10 मिनट वेट चाहिए नहीं आई एम ओके मतलब चाहिए तो इधर क्या बोल रहा है तुम पीछे क्यों गए से
अच्छा रिया ने बॉडी कर दी. Okay, so there is a small exercise. Can you करो ये? Check if you have finished with this. Page 11. Now see, what are very important that you have to definitely go through, learn properly. Page 11 check करो. Page 11, functions of centromere. This we are going to come across now. Now that we are going to study cell division. इसमें ये सारे points we'll have to come across also. But tick करके रखो. What are genes? Definition I already told you. Genes definition you should know. And functions of centromere. Function centromere we are going to come across. Centromere क्या था? What was centromere?
There is centromere is a point of attachment between two sister chromatids.
Two chromatids. Sister name. Two chromatids of a chromosome. That is the point of maximum condensation for a chromosome. Now sister chromatids. Sister chromatids generally duplicated situation में ही उसको sister chromatids बोलेंगे. Okay. So now when we are talking about centromere, that is the point of attachment of the chromatids. Okay, so that is very simple to remember. But उसके और भी क्या functions है that we will come to know when we study. But we are talking about cell cycle. Break के पहले we were talking about cell cycle. There is a small exercise. Check करो. Did you finish that? Page 11 का जो progress check है. Did you finish that? किया है पहले? All of you will try that. If you have done it in school, okay. Those of you who have not done it, you will have to complete that. Complete that. This progress check, page 11. Your book is still the other one, huh? But here for 10th, now see for 9th we have taken care of all the other topics. But 10th में there is no meaning. 10th में only to stick to the syllabus. As it is, there is so much for you to remember. Are they teaching? No, extra चीजें नहीं चाहिए. Then why we are following this book? There's no meaning, no, not necessarily give you that visual stress. Kia? Look at those questions. Chromatin fibers made up of DNA and histones, two sister chromatids of a chromosome attached to each other as centromere. Structure of DNA first discovered by Fleming. Three components of nucleotide. Phosphate नहीं लिखो, sugar नहीं लिखो, pentose sugar, phosphate. DNA strand wound around the histone octamer forms a complex known as nucleosome.
Sugar is also called pentose because it is written sugar and bracket pentose.
Pentose sugar you should always write because sugar तो 6 carbon sugar भी हो सकता है. Hexose भी तो हो सकता है, but DNA में it is never any other form apart from pentose. इसीलिए pentose लिखना must है. A specific sequence of nucleotide on the chromosome constitutes a G. Okay. Now,
while we write the three components of the nucleotide, in the nitrogenous base, do we also write the sub?
नहीं. Only the basic words: nitrogenous bases, pentose sugar, and phosphate group. Now we continue with the concept of cell cycle. Diagram figure 2.8, page 12. Tick karo. Whenever you read this chapter for your exam tests, to take a note, you'll have to read this diagram. Diagram reading is very important. Identification of any particular markings, you should be very clearly able to write. No confusion at all. Now, this was the stages of cell cycle we were talking about. The first one that I said, growth 1 phase or the first growth phase. What are some of the events that takes place? What are some of the events that takes place? I said the cell will definitely increase in size and the volume of the cytoplasm will increase. Then what all the cell organelles will develop, essential proteins, pigments, etc. will develop. Apart from that, some major organelles when we are talking about mitochondria. And if it is a plant cell, then we talk about what? Chloroplast. These two major organelles, they will start the process of division because these two organelles that we are talking about, उधर जा, हाँ उस यहाँ पे तुम दोनों के बिहेवियर मुझे कुछ ज्यादा ठीक नहीं लग रहा है, तो एवरी क्लास बहुत अच्छे बच्चे की तरह यू विल सिट नाइसली टू स्टडी. आया समझ में? उधर जाए. Now, when we are talking about this particular feature, mitochondria and chloroplast. Chloroplast, if it is a plant cell we are talking about, mitochondria, chloroplast, they will duplicate, they will divide. Now, why are we talking about division? Because these two organelles are special organelles. Mitochondria both in the plant and the animal, and chloroplast only in the plant. These are known as semi-autonomous organelles and they can multiply and divide on their own. But based on nucleus, when I have to talk about the basic parts of a cell, for any other function of the cell, which part of the cell regulates? For any other function of the cell, which is that part of the cell which will act as a regulator, which will regulate all the other functions of a cell? That is always the nucleus. The nucleus will always regulate the function of a cell. But here, that one particular point I have written there, you'll have to remember all these features. Mitochondria and chloroplast division takes place in which phase of cell cycle? What are you going to mark? G1 phase. So G1 phase, all these events will take place. Now we talk about the next phase, that is S phase. What will happen in S phase? S from the word synthetic. Synthetic. Synthesis of what? Synthesis of DNA is the major feature here. Synthesis of DNA, meaning by the time the cell has already entered the S phase, the process of duplication has already started. By the time the cell has already, now that the cell is almost voluminous, all the cell organelles are well formed, major cell organelles, duplication also has started. So now the cell will start doubling its chromosome number because it is almost getting ready for the process of division. So synthesis of DNA always to correlate with S phase. Then what next for G2? फिर G2 के लिए क्या बचता है? What are the events that takes place in the growth 2 phase? Growth 2 phase or second growth phase में वही additional proteins and enzymes will be formed. अभी बाकी सारे components cell में form हो चुका है. अभी क्या चाहिए? Next is only those additional proteins and enzymes जो cell division में जरूरत है, that only will be formed here. So what should you understand as a major feature of the G2 phase? Synthesis of proteins. Synthesis of proteins. Now, कौन से proteins? Who can tell me the proteins which are present at the two poles of the cell? Oh, last year you all have learned. Proteins present at the two poles of the cell, जो centrosome में रहता है. What is that protein called? Right? Remember last year we had studied centrosome as an organelle. कौन से protein रहता है?
Ribosomes.
Nobody? Microfilament? Microtubule, microfilament you have studied? Centrioles. Centriole has a particular protein, microfilament. So this is what I am talking about now. अब ये centrosome बनेगा, microtubule बनेगा, because it will help in the process of the spindle formation and etc. So till here the cell is all regular, the process of growth already done. Now here synthesis of proteins and enzymes necessary for division. Only that you should remember. Necessary for division. Division के लिए whatever that is necessary, that only takes place in the G2 phase, growth 2 phase. So this is all about interface that we are talking about. Interface में what is happening? It is also known as the resting phase. Why is it called the resting phase? Because the cell is growing, taking its time to get mature. Basically resting phase, and now it will enter into the process of division. So division के phases क्या है? 4 major phases of division. 4 major substages of division. What are the 4 major substages? I would start with prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, telophase are collective term for all these stages. What will be termed this as karyokinesis. Karyokinesis. So let us now talk about the concept of karyokinesis here. What is karyokinesis and what are some of the stages and events one after the other? So I start with the process of karyokinesis. But how will you define what is karyokinesis? Karyokinesis is the sequence of events that leads to division of the nucleus. Then what is cytokinesis? Division of the cytoplasm. Division of cytoplasm. So what is karyokinesis and cytokinesis? And what are the substages now? The substages of karyokinesis, if I have to talk about the sequential events, that would be prophase. Followed by metaphase, followed by anaphase, and this will be followed by telophase, and a shorter phase, that is cytokinesis. It is not a part of karyokinesis. I will number this as 1, number this as 2. So a difference between these two words, again very common in your exams, in your paper. Differentiate between karyokinesis and cytokinesis. This is what we should be able to understand. Let us now talk about the different stages of karyokinesis. All of you will take your pencils. A basic, the stages of cell division, not the cell cycle. Stages of cell division you should be able to draw, especially mitosis. Meiosis never comes in the exam. Mitosis you are supposed to draw. As in when I am talking about the characteristics, try identifying the stages. With what are the events? Very easy, but still when you see your question paper, children make mistake in identifying the stage. If the stage identified galat hoga, then the rest of the questions sab galat ho jata hai. So your stage identification has to be very clear. So let us start. Just some 6-7 diagrams only you will have to draw. I want all of you take your pencils and we'll have to talk about understanding all these stages. Let us start. The first is, we'll start with prophase. Interface already we were talking about. Okay, if I start with the interface cell. Let me start with the interface cell. I start with the interface cell here. How should you draw? How should you identify? In case of interface cell, already I had mentioned a number of times in an interface, the chromatin fiber would be almost like a ball of wool. Can we identify, can we count the number of chromosomes? No, they are extensively compact, almost intertwined just like a ball of wool. And they will generally not have the nucleolus and all this, nothing of these structures will be well formed. At the most, we may be able to see the centrosome roughly, only that much, nothing else. So centrosome, we can just draw the centrosome. The centrosome only we can draw, that's it. So this is a basic interface cell. If this is an interface cell, then how can we start? How can we differentiate an interface from at the last page of the textbook? I think that's a blank page. Blank page, eh? Yes. So diagrams are proper. The same thing should go to your notebook. In case of a prophase stage, now next after interface, this is the interface. Once you draw the diagram, I'll ask you to label parts later. Now you will not label anything. Now you will not label anything, just you will draw with a pencil. Next is I talk about the prophase stage. Now, this is the nuclear membrane. Prophase stages, there are sub-stages. Early prophase hai, late prophase hai, mid prophase hai, late prophase hai. Let me just draw two diagrams. So initially I would draw that this is a prophase stage. Nuclear membrane is intact. अब ये जो chromatin था, this will start getting condensed and take the shape of distinct chromosomes. What about the centrosome? The centrosome is also well marked. The centrosome is also well marked now. Centrosome is what? I'm talking about centrosome. Centrosome क्या है? It is a structure है, organelle है, pigment है, क्या है?
It's an organelle of the cell.
Animal cells.
It's one of the.
Animal cell का एक prominent cell organelle है. Without this, the process of cell division possible नहीं है. So this is one stage now. Next, you will just draw. You will not label anything. Next, I draw the other stage where the nuclear membrane is disintegrating. This is also a feature of prophase. This is also a feature of prophase. But by that time, what also has happened is the chromosomes have started duplicating. Duplication of the chromosome has happened. Show the chromosomes distinct. By the time the duplication of chromosomes have happened, what now next? The centromere will start its, the centromere also will start its division. Centromere also starts division. So the centromere also will divide. The centromere also will start dividing and it will start moving towards two poles of the cell. The centromere also will start dividing and move towards two poles of the cell. You will practice drawing. At the last page of the book, you will draw. Last page में draw करना. Those of you who don't have the notebooks, you will draw now and see that you do it again in your notebook later. This stages of the diagram, you should be able to draw neat.
Second and third one is the protein.
Yes. I will give you questions. पहले खाली diagram draw करो. Okay. This many diagrams at least you should be able to identify. Let us try understanding the basic characteristics and their identification. Now this particular phase is interphase. How will you identify? This is interphase because if you see the cell is comparatively bigger. How will you differentiate between interphase and daughter cells? See, the last stage when we are talking about daughter cells are comparatively smaller, and as it is daughter cells exam में नहीं आता है. Daughter cell stage is never given in the exam. Interphase stage may come. How will you identify? Interphase stage, chromatin fibers. Label X. What to write? Chromatin fibers, chromatin threads. No? And when I talk about chromatin threads, in interphase is the nuclear membrane intact. Yes, the nuclear membrane is intact. What else now? I talk about this particular stage. What is one distinctive feature of prophase and what is its difference from interphase? How will I understand that this is a prophase? Chromatin fibers have condensed and now they are chromosomes. So mark 1 and mark 2 in this figure. What will you mark 1 and 2 as? One will be what?
Centromere.
Chromatid. Can I mark it as chromosome? No. One is what? One is chromatid. One is chromatid. Then what should I write as 2? What is 2? Centromere. Very clear in the figure, no? Check when you get this type of a question. Very clear. Centromere. So now, how do I make it confirm that this is the prophase stage? Because chromosomes are formed. Chromosomes are formed. But if this is prophase, then what about this? This is also prophase. This is also prophase. It is late prophase. Then in your exam, are you supposed to write late prophase and mid prophase and etc.? Nothing, only prophase. Because now I show you 2-3 diagrams per stage, but in your exam you will get only that one diagram. So anyone, maybe this or this, for example. So tumko, you don't have to mention any late or mid or anything, only prophase to mention in your paper. So this also I will write prophase. Convincing feature kya hai? Chromosomes are distinct. Centrosome is there at one side of the pole, but you have studied that nuclear membrane disintegrates during prophase, but isme to nuclear membrane intact hai. So should I write as prophase? Yes, because chromosomes are distinct. Definitely it is prophase. But what about the other stage? This also is prophase. Both these stages are prophase that I have drawn. Again, I mentioned in your exam, you are not supposed to write early, mid, late, anything. How is this prophase? Because prophase ke features ye bhi hai when chromosome duplicates, that is the prophase stage, or in the prophase stage chromosome duplication happens. What is the other feature of prophase? Centrosome duplication takes place and nuclear membrane disintegrates. So all these are features of prophase. Chromosomes are formed, chromosome duplication, nuclear membrane disintegration, and centrosome duplication. All these are features of prophase. How will I identify this stage as metaphase? Why? Justify this stage is metaphase. All aligned in one particular plane of the cell. What is this plane A known as? Equator or equatorial plane. This is known as equator or equatorial plane or equatorial plate. These are the different words. So if this particular stage, how do I convince that this is definitely the metaphase stage? Because the duplicated chromosomes are all aligned in the equator of the cell. What about the nuclear membrane? No, it's not there. Disintegrated. Then what is substituted by the nuclear membrane? The formation of the spindle. What is spindle? Some protein threads between two poles of the cell. These are known as poles of the cell. Now, if this is marked, mark label A and label B. I have marked here. Inside, if I draw this, it is a structure something like this. This is the cytoplasm. These are some proteins, and these are some protein rays again. I am asking you now, mark this, mark this. A is what? Esters or estral rays. Esters or estral rays. So B क्या है? B is the centriole. ठीक है? So this B is the centriole. What is centriole? Centriole is the protein that is present in the centrosome, which is responsible for forming this spindle. Okay, now this is about the metaphase. What are the distinctive events of metaphase? The duplicated chromosomes in the equator, centrosome at opposite ends of the cell, two distinct poles of the cell, the spindle fibers very distinct. Are the centromeres attached to the spindle fiber? Yes. And the cell is little bit oblong. Cell thoda sa oblong hota hai. After that, now next what? How is this convincing that this is an anaphase stage? इसीलिए, because the chromosomes are already separated, splitting of centromere has happened. So what is you get this type of stages and you'll have to give reason, justify. So what is one distinctive feature of this? Splitting of centromere. Splitting of centromere is one special feature of anaphase. But this is an animal cell or a plant cell? That also is asked. How will you convince that this is an animal cell? Because there is furrow formation. इसको बोलेंगे furrow. जो depression आ जाता है on the cell membrane. That is called the furrow formation. Since we get to see the furrow formation, definitely it is an animal cell. So how to convince that this is an anaphase stage? Because the duplicated chromosomes have started splitting and moving towards opposite poles. It is an animal cell because furrow formation is very distinct. Move on. What is this particular stage? This is also anaphase. ये भी anaphase है. So how it is anaphase? Furrow formation deepening and chromosomes are almost settling at the poles. Chromosomes are almost settling at the poles. So इसको why can't we say that this is telophase?
Because they haven't formed separate nucleus.
Because the nuclear membrane have not
Separated.
reorganized again. Nuclear membrane अगर reorganize हो जाए, then that is telophase. तो यहाँ पे तो nuclear membrane reorganize नहीं हुआ है. Remnants of the spindle fiber is still there. But yes, the chromosomes have settled at opposite poles. What about the centromere, centrosome, opposite poles? What about the furrow, deeper? Furrow formation is very clear. What about this? Cytoplasmic constriction is almost taking place and nuclear membrane has reorganized. Since the nuclear membrane has reorganized, then we have to consider this as telophase. What about this one? Cytoplasmic division complete. These two are daughter cells and the stage name is cytokinesis. The stage name is cytokinesis. So now that we understand that what is karyokinesis, the sequence of events of the nuclear division. Sequence of events, कौन-कौन से events थे? Prophase, metaphase, anaphase, telophase. Sequence of events associated with nuclear division, that is what is known as karyokinesis. And what is cytokinesis then? Cytokinesis is the division of the cytoplasm, which is a longer phase. Karyokinesis is a longer phase. Cytokinesis is always a shorter phase. Which stage has substages? Karyokinesis has substages. Cytokinesis के कोई substages नहीं है. Now look at those set of diagrams for the plants. The plant cells, now you have to identify them. That how to justify that one diagram given in the figure is a plant cell or of an animal cell. And this, अच्छा, plant cell square भी no, not square. You should not write rectangular. Commonly होता है, but sometimes you may get hexagon. These are the features. Square, rectangle ऐसा नहीं लिखना है because hexagonal and plant cells hexagonal भी होता है. So where are the differences? The first difference always you should notice, you should write is if it is an animal, if it is a plant cell, then I should have drawn the cell wall also. Two membranes होना चाहिए.
I mean the telophase plants requirement.
Yeah, let us go sequence. So first feature क्या है? If it is a plant cell, then what are some of the major features? If I talk about a plant cell, on what features you should always write your answer. How to justify that it is a plant cell? Number one, cell wall present है या नहीं, that you will always check. दो membranes है कि नहीं, that you will always check. Two walls mentioned. Shape always don't, generally the rectangular shape is given, but still you always should check two membranes are there or not. Second one, centrosome, centriole there or not. Centrosome, centriole if present, definitely an animal cell, absent, plant cell. Furrow formation if there, furrow formation if there, plant animal cell, and instead of furrow formation now cell plate is there. In case of, instead of furrow formation, take this diagram, page 15, for page 15, check. Cell plate, very clear. Telophase, see that diagram for telophase. These are some of the common diagrams that you will get in your exam. Cell plate formation, cell wall present, and absolute absence of the centrosome. These are the three features on which you have to write your answer. One more question is that the plant cells does not have centrosome, but do they have the spindle? They have a spindle. तो centrosome नहीं है तो spindle कैसे बनेगा? They have got proteins scattered in the nucleoplasm which assemble to result in the formation of the spindle altogether. But the spindle is not formed from the microtubule at all. From the centrosome at all, from the centrioles at all. We will write one question here. Write this question. Plant cells do not possess. This you will always get as a 1 mark question in continuation with some other question. Plant cells do not possess centriole and centrosome. Plant cells do not possess centriole and centrosome. But still, spindle is well formed. Spindle is well formed. Full stop, justify. Full stop, justify. Answer. Plant cells do not have. Plant cells do not have centriole and centrosome, but the spindle is formed due to polymerization. Spindle is formed due to the polymerization of the microtubular proteins. Polymerization of microtubular proteins due to the polymerization of microtubular proteins scattered in the microtubular proteins scattered in the nucleoplasm. Microtubular proteins scattered in the nucleoplasm. Straight away go to page 21 of your textbook and try E sub number 1, 2 and 3. E sub number 1, 2, 3. फटाफट करो. Check which are those questions.
आपने telophase कुछ तो बोला था मैं वो मिस कर दिया.
बताओ क्या?
आपने कुछ cytoplasm
Constriction. Telophase में cytoplasmic constriction will start complete at the cytokinesis stage. Cytokinesis मतलब cytokinesis means cytoplasm division complete है.
And the daughter cells are fully mature.
Fully not mature. They will have few organelles, chromatin fibers, a small amount of cytoplasm. अब ये cell will go through the G1 phase.
So after that it can
Rest.
Yeah, okay.
This cell will now enter into the resting phase.
Okay.
अब होगा G1, S, G2, फिर ये ready like this. फिर ये same continue होता है. करो. ये सब ना को 1, 2, 3 fast. Textbook में करो ना. This chapter is done in your school.
No.
Yes, ma'am. Yes.
Text के questions नहीं किए.
Yeah.
Textbook में लिखा है answers. कहाँ पे लिखा है? Where did you all write these answers? Textbook में नहीं लिखते हो? Where do you write? हाँ? अच्छा. लिखो. Textbook में भी लिख सकते हो. If you want to write in the notebook, you write.
And this mitotic phase.
Plant cells में फिर spindle fibers, microtubular proteins होते हैं.
Just now I gave you this answer. That's why. देखो. लिखा ना? This is an important answer. हाँ.
Mitotic phase.
Mitotic phase. This entire process is known as mitosis. This entire. इसके पहले मैंने explain किया था ना. One cell divides into two cells. So what happens? This is the parent cell, and these two are the daughter cells. If this is the method of division, जहाँ पे one parent cell divides into two daughter cells, this particular method is known as mitosis. Again, I will talk about the differences mitosis, meiosis. देखो what I have explained you just now. If you are able to understand, ये sub number 1, 2, 3.
And in the molecular structure of DNA, certain colors are given like for thymine, cytosine. So color भी important है.
नहीं. वो कलर के हिसाब से कुछ नहीं लिखना है. Like suppose number 3 and number 5. तो number 3 and number 5 क्या लिखना है? नाम लिखना है कि only nitrogenous base लिखना है? सिर्फ nitrogenous base लिखना है.
मैंने
हाँ, pyrimid
और fourth को मैंने bond लिखा. Hydrogen bond.
Hydrogen bond. हाँ. 3 and 4. Purine and pyrimidine you actually cannot write because purine pyrimidine का structure तुम लोगों ने पढ़ा ही नहीं है.
मुझे ये बता रहा है कि purine is bigger, it occupies more space, and pyrimidine is smaller, occupies less space. तो diagram के साथ जो छोटा है मैंने उसको pyrimidine बना दिया, जो बड़ा है pyrimidine.
Actually, that is not the correct concept because structural, though we understand purine is a double chain structure, इसको बड़ा लेते हैं, but जरूरी नहीं कि एग्जाम में हमेशा इस तरह के बड़े छोटे डायग्राम्स नहीं आना. ठीक है, लिखो अभी. But if it is of the almost of the same approximate size, अगर same दिखता है, then better always write nitrogenous base, and hydrogen bond. So number 1. So 1 should be what? The first figure. 1 is phosphate. 2 is very clear. Pentose sugar लिखना है, and the rest 3. How many strands are shown? How many strands are shown here?
1, maybe 2.
कैसे 2?
Like school में करा था यार.
कैसे स्कूल में नहीं मैं पूछूं. I have explained you. I have explained you just now. 2 लिख रहे हो, you should know the reason. 1 लिख रहे हो,
अब 2 रख सकते.
हाँ. So जब I have explained you also, this is one strand connected by hydrogen bases, and this is another strand. So 2 strands है. So these 2 strands are connected by nitrogenous bases. I had explained you know they are known as the ladder. So ladder का structure है the steps known as the rungs. So 2 strands है, very clear. How many nucleotides? 4. है ना? 4 nucleotides. Keep marking there. Name the parts 1, 2, 3, 4, 5. We just now discussed. Name the DNA unit constituted by parts 1, 2, 3. नहीं 1, 2, 3 है ना? Nucleotide. 1, 2, 3 nucleotide. Now this method that is shown, the concept of DNA duplication, where one chromosome duplicates to form two chromosomes. So similarly, the one DNA will duplicate. Now, just now that we talk about a concept of chromosome duplication. If I have to understand, ये जो chromosome के अंदर, what is the chemical composition of chromosome? What is the chemical composition of chromosome? DNA. ये है एक chromosome. This chromosome, when it shows a structure, something like this, what will we call it as? Chromosome duplication. So, इसके अंदर how many DNA molecule? इसके अंदर how many DNA molecule? One DNA molecule and double stranded. तो ये जो DNA molecule है, if I have to understand ये एक से दो बना कैसे? This two DNA molecule. Now, this DNA molecule has got two strands, two threads. These two threads, ये hydrogen bond dissolve हो जाता है and they will separate. So, धीरे-धीरे this one of this parental strand and the other parental strand will come up, separate हो गया. Based on what is the information यहाँ पे, यहाँ पे, यहाँ पे, यहाँ पे. What are the nucleotides? समझो यहाँ पे nucleotide with A है. So which nucleotide should come and get attached here? T. यहाँ पे nucleotide C है. So which nucleotide? G. तो इस तरह से एक नया strand बनेगा. तो इस तरह से एक नया strand ultimately बन जाता है. इस तरह से एक नया strand ultimately बन जाता है. So these are the two chromosomes that I am talking about. DNA duplication. What is the meaning of DNA duplication? From one DNA duplex, two DNA duplexes are formed. From one DNA duplex, I can draw this as a chromosome. I can draw this as a chromosome. Chromosome के अंदर जो DNA है, one DNA split, and based on what is the information in the parental DNA, a new DNA molecule will be formed. This is what is known as DNA duplication. Because next question, the three sketches given below A, B, C are intended to represent. Underline the word replication. Replication and duplication is the same thing. What should be the correct sequence, starting with the first and end with the last? B, C, A. Correct. Complete number 3.
Each chromosome has only one DNA strand.
One DNA molecule. Strand दो.
दो strand,
One molecule. Clear है?
Yes.
Is it clear? No? कौन सा? ये चीज clear नहीं है?
Yeah.
अच्छा. This one is, say, first you see the chromosome. This is one chromosome, like something like this. Clear? I am showing you that how does this chromosome become duplicated? How does this single chromosome become duplicated? How will it become duplicated? For that, we'll have to know the DNA inside. How many DNA molecule inside? One. But that DNA molecule has two strands. So I may draw this DNA molecule something like this. This is the DNA molecule. Already explained in the previous figure that this DNA molecule will definitely have a sequence of nucleotides. Yes, it will have a sequence of nucleotides like this. So it will be all over. It will have some sequence of nucleotides. So whatever is the sequence of nucleotides, based on that the next step. Okay, now the next step is what? The next step is these two strands will separate. How? Because of the presence of a large number of enzymes that is there in the nucleoplasm, these bonds dissolve. तो ये bond जैसे dissolve हो जाएगा, धीरे-धीरे the entire DNA will start splitting up. So for example, I may show a structure like this first. Maybe now I show a structure like this first, where the DNA has partially opened up. The DNA has partially opened up. यहाँ पे अभी भी dissolve होना बाकी है. But यहाँ पे the nucleotide is A, so definitely one nucleotide with T will come. Nucleotide is G, the other nucleotide with C will come, and a bond will be formed.
But how did this T and C come? It's
Not how did this come. These are units already there in the nucleoplasm. Only that the chemical components come together based on their affinity, based on their unstable structure, chemical composition. So now, based on what is the information in the parent strand, I may say that here is C, so definitely now the G will come and इस side में भी एक और DNA synthesize होता है. The process is elaborate. एक side से दोनों DNA synthesize नहीं होता है. But anyways, now this will be the method. So धीरे धीरे what will happen is these two black strands that I am showing you, these are the parental strands, original strands है. तो ये original strands धीरे धीरे separate हो गया. Original strands धीरे धीरे separate हो गया, and based on these original strands, the new strands are formed. Based on these original strands, the new strands are formed. So ये एक chromosome है, this is another chromosome. अगर मैं उसका पूरा structure draw करूँ, तो this is the DNA of one, this is the DNA of the other. आया समझ में? This is the DNA of this, and this is the DNA of this.
And so the original strand determines the characteristics of.
हाँ, the template बोला जाता है उसको. That is known as the original parental template strand. उसपे जो information रहेगा, based on this, new strand will be formed. We'll just very quickly complete with this question. Number 3. Name the parts 1, 2, 3. One is centriole. One is centriole. Number 2 is spindle fiber. Number 3 is chromatid. पूरा structure नहीं है, तो chromosome नहीं लिखेंगे. Chromatid. Identify the verb stage. Anaphase. Mention the type of cell. Mention the type of cell in our body where this type of cell. Mention the type of cell in our body. Somatic cell. Somatic matlab? Yes, I did not discuss about this. Somatic and germinal. दो तरह के cells हैं हमारे body में. So this process that we had studied are the type of cells of the stomach and the liver and the bone and the muscle, but not the cell of the testis and the ovary. ये जो process of mitosis we have discussed today, that is only happening in the skin cells, muscle cells, bone cells, liver cells, cells of the stomach, cells of the intestine, blood vessel, etc., etc. But testis, ovary के cells में ये process नहीं होता है. So what is this process known? What are these cells known as? Somatic cells. We'll complete with this. Mention the type of cells. Somatic लिखना है. Name the stage prior. Metaphase. ये anaphase है. Prior होगा. Metaphase. Succeeding होगा. Telophase. Okay. We'll continue again with the next set of questions and few more topics left for this chapter. We'll continue in the next class. Let's go next class.
17. अभी स्कोर देख ना एक बार. स्कोर देख ना. आप कौन सी टीम को सपोर्ट करते हो? क्या?
सपोर्ट करने हैं तो मुझे ICSE या
135 overs. We will see you later. Aditya sir, my God, SRS versus RR very much. कितने मार रहे हो? SRS
लेकिन आना चाहिए।
240.